Hello everyone, welcome back to another session of RNA biology. So we were here learning about the Epitranscriptome modifiers in three categories: writers, erasers and readers, and we will see their importance with some examples in the coming sections. The N6-methyl adenosine and one of the protein factors that recognize the modified adenosines. They are basically the readers. So the YT521B homology, and we usually call them as YTH domain family of proteins, and there are different members like YTH-DF1, YTH-DF2, YTH-DF3, YTH-DC1, and many more will be discovered in future years, and they have been characterized as the direct M6A, that is modified adenosine bearing RNA, and these proteins recognize these modified adenosine and we call them as readers and have a conserved M6A binding blocks. All proteins have a specific domain in their sequence, in their structure, that will recognize this modified adenosine or methylated adenosine. One example is insulin-like growth factor 2 mRNA binding proteins 1, 2, 3, that is IGF-2BP 1 to 3 are reported as a novel class of M6 readers. They are joining the group. Say for example, many proteins in a system have a bona fide function and where they are supposed to do that particular function. However, many proteins will have an altered functions also. We usually call it as promiscuous function or promiscuity. So a given enzyme or a given protein or a given receptor will have a defined function. However, given a particular circumstance, it may perform a different task. And one such example is the IGF-2 mRNA binding proteins. So they have their own dedicated function. However, they can recognize some specific modified RNAs as well. IGF-2BPs use a K homology, that is KH domain, that is K stands for lysine, H stands for histidine domains, to selectively recognize M6A containing RNAs and promote their translation and stability. In general, we can say as a thumb rule, if an RNA have got this M6A adenosine residues, they will facilitate, they will be marked for a translation and they will be marked for their stability. Means if an RNA has modifications, this methylation of the adenosine residues, they will be marked for their stability. Means you can simply say prosperity of an mRNA. What is the function of an mRNA? It has to be stable and it should be available for translation. Both it is doing. It's almost like giving a tuition or special class to a student. That means it will boost up the RNA's function. It may sound okay, it may sound great. However, if this RNA or the product of this RNA, that is a given protein, is not so welcome or not so much needed in a given scenario, that can lead to problematic situations. Just like obesity, you think about it, like if you are eating a lot and your body is not wasting anything, it is depositing in your body. Isn't it a fantastic thing? But is it really so that you have a lot of fat deposited in your body? Is it a good thing? It is definitely not a good thing, because food need not necessarily be stored in your body forever. It has to be stored for managing an emergency. Say you did not eat one day, could not eat in time, so your body's glucose should not dip. So it should simply be able to manage maybe for one day. But some people will have food stored in their body for 2 months or even 6 months. So that is not a healthy sign. Similarly, RNA facilitated by this kind of external factors, in some circumstances it is beneficial. but in some circumstance it will accelerate, it will boost up. So it's like pouring oil in a fire. So this cage domain is selectively meant for recognizing the modified RNAs. So these M6A readers together with M6A methyltransferase, that means they are the writers, and demethylases, erasers, establish a complex mechanism of M6A regulation in which the writers and erasers determine the distributions of M6A on a given RNA. Which are the A's should be modified, how much dense M6A should be present in a given RNA has to be decided by these three, that is readers, writers and erasers. Whereas the readers mediate M6A dependent functions in general. So M6A also has been shown to mediate a structural switch termed as M6A switch. That means it can influence the stability of the RNA due to its unique structural properties. So the differential M6A profile is an emerging hallmark of cancer. So that is why this modification becomes very important. You can predict, you can detect whether this particular tissue is vulnerable to get cancerous or not purely by looking at the M6A profiling of certain RNAs. Just like you may have heard about measuring your glucose, fasting glucose level or glycated hemoglobin levels, etc. as an indicator of diabetes. There is also possibility of predicting a potential cancer in future. So aberrant M6A deposition plays a key role in tumorigenesis in various cancers by broadly altering the gene expression. So by and large, the tumorigenesis is occurring because of an accelerated stability and accelerated translation. So this can, just like you think about it, none of the humans are dying from the planet. Every human, some human beings have 500 years, another 1000 years, another 2000 years. You can imagine how much crowded the planet will be, because clearing every species after a certain age is a must for the propagation of the species, not propagation of an individual or propagation of one particular person is not necessary, because maintaining a body or maintaining a particular individual is not desirable for the planet or as a species. It's always good to contribute to the next generation. Why? Because a person who lives longer, just like an RNA living longer, can do lots of undesirable effect, because none of these molecules like an RNA or none of the organism is perfect. It is always requiring change. So dynamics nature of RNA, dynamics nature of individual members of a species is a must. So M6A methylation plays an important role in a wide variety of biological and pathological processes, and including the formation of cancer. One example, one of the M6A methyltransferase METTL3, and it's a M6 methyltransferase is upregulated in gastric, colorectal and liver cancers. So a lot of studies have shown that they can undergo solid upregulation. A drastic upregulation means if you have an elevated action of this methyltransferase, it can definitely lead to cancer, and it is detected so in a variety of cancerous condition, and the presence of this methyltransferase will naturally increase the methylation levels of various RNAs, including non-coding RNAs. The M6A modification frequently involve key components of oncogenic pathways such as Wnt beta-catenin pathway, PI3K AKT mTOR signaling pathway. So these are all pathways. Say for example, the Wnt beta-catenin pathway is a must. Like in order to repair your lost cells, like your hair is lost, the Wnt signaling is important to rejuvenate your new hair growth. Say your skin is torn, or you got a cut in your hand, or you got a mosquito bite and your tissue is damaged, you got a pimple and you made some tissue is damaged. So you need to repair that damage. So starting from the minutest damage occurring in your body to persistent damage like a big wound, a blister, a wound that formed because of fire accident, or even your intestinal epithelium constantly, because of the movement of post-digested food particle, your intestinal epithelium is constantly doing a lot of wear and tear. So all this repair require Wnt signaling. So in a nutshell, you can say without Wnt beta-catenin signaling, no organisms can survive. Organisms' chance of living is zero. So it is so important. However, the same signaling is super upregulated during cancer. That means there is a fine balance between not having Wnt signaling and excessive Wnt signaling. Both are bad. Not having Wnt signaling also is bad. Too much of Wnt signaling also is bad. So you have to live in between. So M6A modification accelerates the Wnt signaling because it stabilizes each and every component of the Wnt signaling. So naturally, the Wnt signaling gets deregulated into a positive side. That means accelerated side. So this can naturally lead to cancer. Same way you have this PA3-AKT-mTOR pathway. So which is basically a cell proliferation associated pathway. That means if you have AKT upregulated, you will end up having too much of cell proliferation. It is meant for maintaining the homeostasis. Basically, system has to decide, system has to sense whether a given tissue or a given tissue should proliferate or should not proliferate. Both these things are decided by the Wnt beta-catenin pathway and the PA3-AKT pathway. So both these things are upregulated drastically when it comes to the M6A superactivation or M6A methyltransferase is upregulated. And both these pathways, when they are superactive, that eventually contribute to their apparent activation leading to cancer. So understanding the function and molecular mechanism of M6A mediated epitranscriptome may thus unravel the novel therapeutic targets and biomarkers for the cancers in early detection. So it is important for us to know whether a given tissue is vulnerable to get cancer in future. That is one way. These days we have lots of ways of detecting it by genotyping. Like many people you may have heard about it, some famous celebrities who are vulnerable to get cancer, they get rid of their tissue, amputate much before that so that they can escape from the cancer. That is a proactive measurement. And some other similar approach is, although your genes don't have any mutation, then how are you going to detect it? Because genes are perfectly fine. The RNAs that is formed is now undergoing this kind of changes. Then you should have a ways of detecting or ways of measuring such an altered RNA is present or methylated adenosine bearing RNAs are present. So this is also helpful in advising a person or a subject whether they are vulnerable for cancer in a given tissue. So lot of applications can be attributed to such kind of studies. So let us think about an example. Multifaceted role of YTHDF1 in gastrointestinal cancer. Gastrointestinal cancer is very complex and many times it is extremely invasive and hardly any treatment can be done because they are so aggressive. By the time you detected the gastrointestinal cancer, the time may be almost over. Means you detected once the cancer has established itself. That is why it becomes so aggressive. They remain silent. They do not give any clue. They do not give any indication. By the time you detected it, it is way too late and no possibilities of recovering it. So that is why gastrointestinal cancer becomes so pivotal. So let us see. You have a methylated RNA. It is sensed by the YTHDF1 because it is a reader and it can work along with some of the signaling pathway like we have mentioned here, Wnt beta-catenin signaling pathway. That is Wnt beta-catenin, I will briefly mention. Wnt is the ligand and the beta-catenin is the product, and the product is normally being formed, that is the beta-catenin in a given cell. However, the product is not stabilized. The product is marked for degradation. However, if the wind is present in the system, in the environment, it binds onto its receptor, that is the frizzled receptor. It binds onto it and helps in stabilizing the beta-catenin. The stabilized beta-catenin goes to the nucleus and help in the transcription pathway, which can be usually the cell proliferation, and new cells are formed and it can repair the tissue. It is pretty simple. Many components contribute to it. We will not go into the details because there are like GSK3 beta is there, like disheveled, many proteins are there. But idea is quite clear that if you have a stabilized beta-catenin in your system, it can facilitate proliferation. It can be regular proliferation or cancerous proliferation. So, lot of proteins in this mRNA, lot of proteins comes in this category. Like you can see AR, HGEF2 and USP14, FCD5, CMYC, FCD7, so on and so forth. Many of them are components of the Wnt beta-catenin signaling pathway, and eventually you will see an accelerated Wnt beta-catenin, AKT, GSK3 beta, Rho-A pathways, and it will lead to tumorigenesis and metastasis. Metastasis means we already kind of discussed that this cancer cell originated from a given tissue, established a secondary or tertiary colony in some other organ and damaging that organ also. So that becomes a, doctors usually say stage 1. Stage 1 is the beginning stage, stage 2, stage 3, stage 4. Stage 4 is the last stage. So when the secondary has spread all over the body. Another example is that FCD9, TCF4, DVL3, FCD7, TED1, these proteins have this methylation and it is detected by the YTH domain family containing protein, that is YTHDF means YTH domain family protein. So they can stabilize the Wnt beta-catenin and can lead to stemness and also drug resistance in case of various cancers. Stemness here what we are referring to like cancer stem cell. So normally when you do chemotherapy in case of a cancer, many times cancer will completely disappear, everything is perfectly fine, cancer cell, the tissue damaged, everything is fine. So after a few months, then again they will start showing the signs. So that is because of the presence of cancer stem cell. Why they did not die earlier? Because they were not showing any characteristics of cancer. So it is just like, it's like a normal cell. So not even proliferating. Normally all chemotherapy targets those cells which are proliferating. If non-proliferating cells are also dead, then the organism will be dead. After chemotherapy, the patient has to be cremated. So that you don't want to happen. So chemotherapy, when you are treating, always targets proliferating cells. But these cancer stem cells still remain dormant. They do nothing. So these drugs can do nothing onto them. They will come back to cancer stage later. So when they come second time, they are much more experienced with the chemicals and they are more fit than the old cancer condition. So these stem cells, when they come back, they will completely dominate and the patient will be dead in no time. So that's why cancer stem cells also a problematic situation, and that lead to drug resistance because they survived earlier drug treatment. Next time you can't use the same drug. If you use that, the cells will be very resistant even while they are proliferating. So another list of genes like ATG2A, ATG14, B, C, and so you don't worry too much about the names of the gene. These lists are included mainly because to understand how diverse the number of mRNAs we have and how they are contributing to it. Like these pathway genes, they lead to autophagy and ferroapoptosis. So this happens because there will be an elevated metabolism and running short of resources, and this will lead to autophagy. Autophagy means self-eating. Like simplistic way, you are very hungry, no one is giving you food, so you are eating your own finger or you are eating your own body's flesh. How does it sound like? Like you have no food, no one is giving you food, so you are biting your own body and eating. So this is what cells will do, and they want resources and they don't have resources available, not available from outside because they are utilizing too much of resources. So they will do eating up themselves. And our body does always, like you don't realize it. Like if you haven't done a long running, if you haven't done lots of exercise in the gym or something, or after 6 months of gap or after 2 years of gap you are going to gym and you are doing some exercise. So what will happen? After 2 days you will have all over your body, you will have pain. Earlier you used to do same exercise, you had no problem, and then you gave a gap and then you are going, and next day you will have lot of, and that is called, we will say, sour muscle. Why muscles turn sour? Because muscle was able to handle the show 6 months ago when you were actively doing. Now it is not able to handle. What happened to the muscle? Because body ate it up. Because lot of ate the muscle doesn't mean that the muscle went missing. Muscles are made of cells, and the body did not make fresh muscle fibers in your body. The existing muscle, the number of protein, the amount of protein that contributes to the bulk. That is why bodybuilders have got very fat, real bulky body, whereas regular person have got less. This doesn't mean that they have more cells. They have the same number of cells, but the amount of protein deposited per muscle fiber is much high. They also will have, say, 1000 muscle fiber. You also have same 1000 muscle fiber, but theirs is looking much bulkier and stronger because individual muscle fibers have got much more protein. So when you are not doing exercise, your body is utilizing those protein for body's own survival. Body don't want to waste unnecessarily build the protein. That is why first time when you do, this existing muscle is not strong enough and you create micro cracks, micro tears in your muscle, and it takes time to repair. And after 1 week, you have a new muscle. You have new muscle in the sense new proteins in the muscle, and it will be stronger. And if you keep doing it, it will be stronger and stronger. And you keep continue to do, it will become bulkier and bulkier. And this is how normally every body functions. Any job when you are doing within limit. This doesn't mean that 24 hours you are in gym, you will become very bulky. You will be hospitalized. That is what is going to happen. So there is a limit. Your body needs a recovering time. So your body automatically does the autophagy, but it should not be doing simply because of an aberrant mechanism or aberrant. Then what will happen? Some of the crucial tissues will go missing. So your body will start suffering really badly. So that is why autophagy and ferroptosis, that means the elevated metabolism will create a little bit more metabolic temperature, and that also lead to apoptosis, because beyond certain limit, if the cell's temperature goes up, the lot of proteins go bad, then that will be marked for apoptosis. So this is what happens because of this accelerated methylation. And then lot of other proteins are there. I am not going to read the names of each of them because it's not required, and it is helpful in preventing the bacterial infection and inflammation. But these proteins, when they are hyperactive because of their methylation, you will be vulnerable for bacterial infection and even inflammation. Lot of gastrointestinal cancers come because of your intestinal bacteria is producing some of the toxins and your body is inflamed. Inflammation is a trigger to cause cancer. So your body should not get inflamed just because of having bacteria, but it do happen. It can happen because of some of the altered expression rate of some of the mRNA because of this kind of modification. Another example is the TGF-beta, transformation growth factor beta. So this is important in various pathways. Normally it is a negative regulator of cell proliferation, but this is also influential in inhibiting some of the unwanted activation of your immune system, immune cells. So the lack of proper regulation of this TGF-beta in the desired level, that can even lead to some of the autoimmune disorders. Then comes the famous JAK-STAT pathway, which is normally a contact-based recognition system. In the sense, if JAK is activated, it will phosphorylate the STAT, and the phosphorylated STAT goes to the nucleus, and it's a transcription factor. It can create the cell proliferation environment. The JAK-STAT and TGF-beta pathway are kind of negative regulators on when it comes to the immune system maturation, immune system. So the overaction of these pathway genes, you will have a compromised immunity, and it can lead to a lot of complications. So let us see the YTH domain containing readers, and these facilitate the translation of the modified target genes, and this will confer malignant phenotypes and causing cancer cells intrinsic properties such as proliferation, metastasis, and stemness and cell death. So all these things are attributed together because of one common factor, that is accelerated methylation, as well as the interaction with the tumor microenvironment such as inflammation and immunity. That means cancer is already facilitated because of the overaction of many of these genes. On top of that, the body lacks the ability to interact or counter the bacterial toxins, which will facilitate the inflammation and compromise the immunity. In that situation, it's just like someone is about to fall and you are giving a push to that person in the same direction where he is falling. So, falling is assured. So, everything is working together to cause a cancerous condition in several actively methylated mRNA transcripts. So, we will learn more about this RNA modification and their impact in next class. Thank you.